This is our stop-motion film on protein synthesis. It begins in the nucleus where DNA exists in a tightly wound double helix structure, making it compact and manageable. Transcription is initiated when transcription factors bind to specific DNA sequences called promoters located upstream of the gene. RNA polymerase then attaches to the promoter region, unwinding the DNA to expose the template strand. The RNA polymerase continues to unwind the DNA until it reaches the termination site. Zooming in on this process, RNA polymerase is moving along the DNA template strand, synthesizing a complementary RNA strand by adding nucleotides in the 5' to 3' direction. This process, known as elongation, continues until the entire gene is transcribed. Transcription ends when RNA polymerase reaches a terminator sequence on the DNA, causing the newly synthesized RNA strand to be released from the DNA template. RNA processing is essential to produce properly modified RNA molecules that are functional and stable, ensuring accurate gene expression. It includes adding a 5' cap for protection and translation initiation, removing introns and splicing exons together to form a continuous coding sequence, and adding a tail at the 3' end made of adenine nucleotides to enhance RNA stability and export from the nucleus. Spliceosomes, which are complexes of proteins and small nuclear RNA, are responsible for removing introns and splicing the exons together. They recognize specific sequences at the intron-exon boundaries, cut out the introns, and join together the exons to form a continuous coding sequence. A couple key splicing techniques include exon skipping, where the RNA polymerase skips over faulty or misaligned exons, and mutually exclusive exon splicing, where only one of two or more adjacent exons is included in the mRNA. This makes it mutually exclusive, since the inclusion of one exon prevents the inclusion of another. Translation begins with the assembly of the ribosome complex, which includes messenger RNA, transfer RNA, and ribosomal RNA. The mRNA provides the template, the tRNA brings amino acids, and the rRNA forms the core of the ribosome. tRNA first binds to the ribosomal RNA's amino acid or A site. This is the site where the tRNA receives the growing polypeptide chain and adds its own amino acid. The peptidyl or P site holds the tRNA carrying the growing polypeptide chain. The ribosome catalyzes the formation of peptide bonds between amino acids. The exit or E site is where the tRNA exits the ribosome complex. As each tRNA brings its amino acid to the complex, it catalyzes the formation of a peptide bond between the amino acids, linking them together. The complex moves along the mRNA strand, reading the sequence of codons. The tRNA has an anticodon that is complementary to the mRNA codon, ensuring the correct amino acid is added to the growing polypeptide chain.
The ribosome shifts along the mRNA strand by one codon, allowing the next tRNA to bring in the corresponding amino acid. Once the peptide bond is formed, the tRNA in the A site of the rRNA now carries the growing polypeptide chain. The ribosome, after being facilitated by elongation factors and energized by GTP, then shifts down a codon along the mRNA. The tRNA that was in the P site moves to the E site and is released from the ribosome. The ribosome continues to read the next codons on the mRNA, and the cycle of amino acid addition and peptide bond formation repeats. Termination occurs when the rRNA reaches a stop codon on the mRNA. UAA, UAG, and UGA are stop codons, meaning they don't code for any amino acids. Instead, they signal the end of translation. These stop codons attract a release factor protein that recognizes the codon and binds to the ribosome, triggering the hydrolysis of the bond between the polypeptide chain and the tRNA in the P site. This releases the new synthesized polypeptide from the ribosome, and the subunits, mRNA, and release factor proteins can be reused for another round of translation. Post-translational modification. There are many ways a protein can be modified after translation. One way is where the sugars, lipids, or phosphate groups attach to the amino acids. Another way is when the enzymes remove one or more amino acids from the leading amino end of the polypeptide chain. Another way is when the polypeptide chain is enzymatically cleaved into two or more pieces. Finally, two or more polypeptides that are synthesized separately can come together if the protein has quaternary structure. From this cytosol, a free ribosome translates an mRNA molecule. The polypeptides of proteins destined for the ER or secretion are marked by a signal peptide, which targets the protein to the ER. The signal is about 20 amino acids and is recognized by a protein RNA complex called the signal recognition particle. This particle binds to a receptor protein in the ER membrane, and the polypeptide synthesis continues as the growing polypeptide goes into the ER lumen via a protein pore. The signal peptide is then removed by an enzyme, and the completed polypeptide is sent for secretion or other cellular locations.